So I want to start out by thanking the organizers and the people that give us this space to share, I think, very exciting science. I think we need more of this, and in particular, we really need more mathematics injected into what we traditionally think of as biology. We've had several talks that sort of span the gambit already, and I missed some of them on the early end. So suffice it to say, I sort of break this into two different categories. One where when you have more information available to you through complex systems, you necessarily need more sophisticated ways to analyze data and infer, something from the data, biological meaning. And so that tool building is more on the inferential or statistical side. And then there's another part of it, which is modeling complex systems is difficult because everything matters, and there's a lot of dimensions upon which something can fluctuate or dynamics depend. And so that sort of also lends itself very naturally to sophisticated models that are bottom-up models. And I think this piece is something that maybe is not as immediately obvious that we need. I'm very happy that this group is sort of sharing and at least understanding that need. And so today, a majority of my talk is going to be focused on that latter part where you sort of take a ground-up model. There will be some inference that we do using those models. And this is specifically going to be in the context of studying the tumor and immune interaction, and specifically the interaction of tumors with an adaptive immune repertoire of T cells. So, so at the end of this talk, there's going to be kind of a segue into my pedagogical talk, which, depending on time, we'll see how we want to kind of divide and conquer that. I don't want to get in the way of the chai break because I think so far some of the most exciting science has been happening there, and I want to continue that. But the structure of this talk is going to be a little bit of introduction on T cells and why these sort of are important and how we think about them mechanistically as being important at a systems level for tumor immunology. And then I'll go into sort of two broad parts. The first part will talk about our population dynamical models and then biophysically inspired models to understand T cell antigen specificity. So why that'll be important should become clear in a moment. And then in the second part, I'd really like to talk about population dynamical modeling of the tumor adaptive immune interaction. This is a complicated interaction because tumors evolve. The adaptive immune system is not fixed. So in principle, you can have an adaptive immune system recognize a tumor sort of in time in a way where the tumor is, in a sense, co-evolving, trying to escape the immune system, and one of them wins potentially. Maybe they tie. So those dynamics are complex. So that's sort of the motivation for the last part. Okay, so just some very brief background. So there's a lot of hallmarks of cancer. I think we've seen some of a similar version of this slide somewhere. From the standpoint of this talk, really what I want to focus on is this key sort of necessity that in order for a tumor to survive and cause problems, it has to necessarily avoid immune targeting and destruction. And so, okay, this lagged a little bit. So, okay, so this really has a story that starts with T cells. And okay, so what are T cells? So T cells are part of the adaptive immune system. And really their purpose is to essentially recognize a threat that has never been seen before, adapt or control that threat, clearing it from the body, and then develop a memory of that threat so that were it to arrive again, it would be capably cleared in the future. And the key to this, at least on the T cells, is through this molecule that's sort of idealized here in the cartoon, or maybe even in my blob here of a cartoon, is a T cell receptor, or TCR for short. So every T cell has this T cell receptor, and actually part of what makes this an interesting problem is that in a healthy individual, you have on the order of 10 to the 8th of even more, sometimes 10 to the 9th, of these unique TCR clones, each with a different T cell receptor. And they basically go around the body surveying signatures that are presented on the surface of an antigen-presenting cell, for example. Those signatures are digested peptide fragments. In the case that I'd like to focus on, which is the CD8 positive restriction, these are like 9 to 11 amino acid peptides. And they're digested and then they're presented on this MHC, major histocompatibility molecule here, to the T cell. And the T cell has to bind this and make a decision. Does this look like a foreign peptide, non-self? If it does, then the T cell can attack. And if it looks like self, maybe the T cell goes on its way and tries to find something else. So this is really, in large part, used to confer durable immunity against intracellular pathogens, classically like viruses and intracellular bacteria. Diversity of the antigens, again, is about on the order of 20 to the 10th, because you have 20 amino acids and you get on the order of 10 mers. And so really quickly you see, and in fact, this number here, 10 to the 8th, is really for one human. The upper theoretic level of T cell antigenic or T cell receptor diversity is huge. It's something like 10 to the 19. So very quickly we realized that this is a very, I don't know, complex, difficult to characterize problem if we only look at experiments and we don't develop some theoretic insight here. Okay, so this is a picture that basically illustrates a T cell attacking on the right here and attacking some presumably infected, virally infected cell. And again, this is really seen as if the antigen that's loaded on here looks foreign to the T cell and the T cell receives sufficient sort of warning signals, then this attack can happen and it sends a killing signal to the cell. So for the sake of this talk though, it also turns out that T cells do the same thing to recognize tumor antigens. Is there a question? It's an immune synapse that forms with, the question is it like a single one of these T cell receptors recognizes the peptide-bound MHC? In general, there are thousands, actually more than thousands, probably 10,000 of these MHC molecules that are presenting various peptides. And the T cells really are trying to bind this in multiple ways to sort of hone in. Yeah, okay. So presumably though, so this is a simplification if you're only thinking of a one peptide, one MHC molecule, but you would build into a model like this something where you sort of evaluate if there's a sufficient concentration of this molecule being expressed, then adequately presented, and then of course binding to the TCR. So in the same way, T cells can recognize tumors via tumor-associated antigens that are presented on tumor cell surfaces. And these can come in various flavors. There's, if we compare ideally to the self-peptide that I just kind of selected, it might be that during the mutational process of cancer, you end up with a point-mutated neoantigen, a new antigen that didn't exist until the mutational process created it. And presumably this single amino acid difference from self may be a signature that could be recognized, although it looks very close to self. There are other mutations. I mean, other types of mutations can cause more abrupt changes to these neoantigens. Another form of this would be if a self-peptide is overexpressed or mislocalized in a context that under normal circumstances T cells aren't really seeing. And so although that's a self-peptide, the immune system hasn't been trained to avoid recognizing that self-peptide. And so they're really kind of seen as foreign because they look nothing like a presumably tolerized self-peptide. So we kind of organize our thinking into these broad categories. And really the idea here is that T-cell immunotherapies are strategies that are aimed to enhance precisely that killing, that anti-tumor killing effect that was just shown before. And there are various examples. So these three kind of span, in some sense, some of the popular ways of doing things. So tumor antigen vaccines are essentially taking and sampling and trying to predict which tumor-associated antigens would be immunogenic, and then injecting them with the hope that you stimulate T cells that are specific in a human with cancer. That's one way of doing things, very similar to a typical vaccine, right? Just using personalized antigens for that person, that individual. Another one that's a little bit more, maybe a little bit more sci-fi, is this chimeric antigen receptor or CAR T therapy. So here the idea is you genetically engineer a T cell receptor with known specificity to a tumor-associated antigen. And then you insert this, you genetically engineer it, you insert it into the patient with the hope that this is going to hone specifically onto tumors and then avoid reacting with other sort of off-target effects. Now this is a very specific therapy that involves one or a small collection of specificities that you target. Another class that's actually kind of at the opposite end of the spectrum is this hematopoietic stem cell transplant. So in liquid tumors in leukemias, it's very common, for example, that in treating the cancer by giving ablative chemotherapy to the bone marrow, you end up immune depleted. And so there's the transplant of a donor-derived immune system that ends up kind of, as a nice consequence, bringing along donor-derived or allogeneic T cells. And those T cells are different than the T cells that presumably failed in the first place to recognize the tumor that's now being attempted to be controlled in the clinic. And so, in contrast with one or two T cells, really this kind of therapy represents an entirely new T cell repertoire being thrown into the cancer. And this is a very invasive therapy and only used for certain specific cancers. Yes. Yes. Yes.
You're trying to say, from what I understood, was are you trying to create new T cells there after some kind of radiation?
The bone marrow is where hematopoietic progenitor cells arise, and among those are T cells and B cells. And so that bone marrow sort of gives you the ability to develop immunity.
Yeah.
And if you remove it with chemotherapy, because the cancer that you're targeting is also being kind of proliferated in the bone marrow, you also sacrifice the ability to, you become very immunosuppressed because you now have no bone marrow and you have no way of generating new T cells.
But then there is bone marrow transplant.
That's this. Hematopoietic stem cell transplant. Yeah. Is it clear? Yeah, actually, just as an aside, so I don't remember what year they started doing this, but in leukemia, they actually started, right? They said, we're going to give chemoablative therapy. Patients were recurring, and they said, this isn't a good thing. So, we need to give this transplant, and they were dying of other diseases. Opportunistic infections. This was not a good thing. They were completely immunocompromised. They gave this transplant, and the patients were doing better, but oftentimes patients suffered from graft versus host disease, which is essentially the recognition, the inappropriate or undesired recognition of the donor-derived T cells against most commonly, single nucleotide polymorphism differences between, or immunohistocompatibility differences between the donor and the host. So even if we share a same MHC molecule that presents the antigen similarly, which is actually almost a necessary condition for being able to even undergo this transplant. Without that, you have very severe graft versus host disease. You still have minor differences because your immune system was trained to avoid things that looked ever so slightly different from mine and yours, and so on and so forth. And so they actually, and this can be life-threatening too. So clinicians said, okay, well, we have an idea. We'll just remove the T cells because the T cells are the thing that creates this graft versus host disease. And when they removed the T cells, they found that the patient's survival went back to what it was before they even gave the bone marrow transplant. And so this was sort of proof positive that the T cells were actually conferring some benefit in creating durable remission. Yeah. And this has a success rate, I think in AML, a little bit over 50%, maybe 60%. It sort of depends on how similar these immune systems are. Identical twins that are given transplants actually don't do well. You don't want your identical twins T cell repertoire because it looks a lot like your T cell repertoire. On the other hand, you don't want to complete strangers because you'll die of graft versus host disease. So there's sort of an optimal balance that needs to be struck. Yes. Yes. I don't know the answer to that. Yeah, in a sense, the issue here is that the T cell clones that you're creating in doing this are coming from common progenitors, and those T cell clones undergo peripheral and central tolerance, and that peripheral and central tolerance looks different from one person to the next. And so, yeah, so I don't know the answer to that question. It's an interesting thought. But yeah, there's actually a lot of new strategies to optimize this that are coming out where, I mean, they're actually being computationally driven by essentially trying to predict which minor histocompatibility differences exist, and then trying to find an optimal pairing given that, that difference. And those differences can be tens to hundreds of thousands of minor histocompatibility antigens. So it is a very interesting computational problem, and I think an opportunity for mathematics to sort of streamline. Okay, so this is the sort of the introduction, and now I'm going to sort of go into this this part of the talk where I'd like to talk about how we think of this from a statistical physics sort of perspective. If we're trying to idealize these interactions and asking questions about specificity, we know there are a lot of T cells, a lot. We know there are a lot of antigens, I mean, prohibitive numbers to do this sort of brute force. And so can we come up with sort of a population dynamical model of T cell selection that can explain, the recognition or estimate recognition rates of things like tumor-associated antigens that may either look very close to self or not. And so I'd like to just motivate this very briefly by mentioning that there are multiple ways in which T cells get selected for, and there's a central tolerance procedure where developing T cells, these are thymocytes or T cell precursors, make their way to this organ that's in sort of the, I think, anterior chest, called the thymus. And these T cells will undergo selection. So selection happens in two, we sort of envision it as statistically independent steps. The first step is positive selection, wherein a T cell with a receptor is encountering antigen, and the hope is that the T cell receptor can adequately interface with the peptide bound on an MHC. And if it can do that, in positive selection, it receives a survival signal, and it's allowed to move on. So T cells get a lot of opportunities to do this, and if they can't bind, they get deleted. The process of generating T cell receptors is stochastic. There's actually a very cool recombination event that sort of generates a random array of these. And so presumably the first step is we want this T cell receptor to be functional. We want it to be able to interface. And yeah, the survival rates of T cells going through this step is something, I mean, it's estimated to be about 5 to 10% of T cells go through and survive positive selection. So on the other end of positive selection, we have now a T cell that can interface properly. It can interrogate peptide. And now we really want to make sure that peptide or that T cell is not going to recognize self-peptides that are sort of ubiquitously expressed. So these thymic medullary cells start to express a wide variety of proteins that are expressed in sort of distinct organ compartments. So they might be expressing things that are in the skin, in the lung, all sorts of things are really actively being presented to T cells. And the idea is that the T cell now has to make its way through interacting with all of these medullary thymic medullary epithelial cells. And if they bind any event, they're autoreactive and they need to be deleted. So they'd be sent a killing signal. So T cells that come out of all of this are ones that successfully have a TCR that can bind, but don't bind any self-signatures to where they would cause problems. And our question, our motivating question was, how does this process affect the ability to recognize things like tumor-associated antigens where maybe some of those tumor-associated antigens were tolerized against in this particular step? And I just want to mention this central tolerance is only part of the story. There's also peripheral tolerance mechanisms that play equally, if not a larger role in sculpting T cell specificity. But similarly, peripheral tolerance would also, sort of discourage T cells from recognizing things. And so is sort of on the repertoire level, if you will, how does tolerance against self impair or affect your ability quantitatively to recognize a foreign signature? And so that was our question. And our approach was to develop, yes?
So in the thymus, so then the idea is it somehow manages to have the repertoire of self-peptides.
Yes, a repertoire of self-peptides. A subset. It's a proper subset. It's sparse. So it's not a lot. I mean, I thought of as key, important signatures that you really want to avoid. The rest is done peripherally. Once the T cells get to the organ compartment, And then, okay, dynamically they're in an environment that may be pro-inflammatory, maybe it's non-inflammatory, and those signals are more complicated, but they can allow a T cell that passed through these checkpoints to essentially identify something but then not react against it. Yeah. And problems with this are associated with autoimmune issues. In fact, it's very curious that probably the dynamics of the antigen presentation matter, although I think it's still hard to fully understand. During infant development and puberty are a lot of times when you have autoimmune issues sort of arise, and these happen to be places where there's a lot of turnover in the expression of proteins. But that's only sort of indirect evidence that perhaps the precise dynamics of T cell engagement in these organ components matters. So yeah, any other? Yes.
So does this also evolve with time? So the thymus selection that happens, the cell peptide present in the thymus also, say, evolve like as you age?
The fact that T cells go through this process dramatically evolves as a function of age. I don't know if anybody knows whether or not the antigen compartment itself is evolving. There's no, to my knowledge, feedback on what's going on in the body to, okay, we now need to change the presentation in the thymus. But this process is operant usually until somebody hits their mid-20s. And then there's a precipitous decline in thymic output. It's basically the thymic becomes anergic. So the T cell repertoire that you have is sort of maximal. And then you have minimal new T cells arriving, but you still have a little bit. I think it's on the order of 2 orders of magnitude decline. And then as you age, your diversity also begins to shrink. And so I'll talk about some modeling that we've used physiologic declines in T cell turnover and diversity to explain tumor incidence. But that's a very good question. So dynamically, this can change, at least as a function of what we're thinking about for adaptive immune systems against tumors. Okay, so now let's go into the math model. So really and there are previous models, actually digit string models developed by Alan Perlson, who started this earlier, and Arup Chakrabarti also had similar out of MIT, similar models of engagement. And so we present our variation of this sort of random energy type of modeling. So the idea is that the TCR can be sort of subdivided into 2 parts in their interactions involving this peptide. One interaction involves the TCR with the MHC. So the MHC, for an MHC restriction that we fixed, is not going to change. We have 6 copies of this, but assuming that we're focused on one individual's immune system and we're looking at one of these copies, the T cells that we're describing are largely going to be seeing the same MHC molecule. Furthermore, as I told you, the T cell receptors are capable of random generation, and so a lot of diversity in their production. But the part of the TCRs that are very diverse are not in this part of the TCR that touches the MHC. So these are called CDR1 and 2 regions, and they're distinct from this other more variable region called the CDR3 region of a T cell receptor. So it's this variable region, hypervariable region, together with the fact that there's antigen that's also a variable that basically contributes to a variable interaction. And we can think about this interaction as sort of an energy. So we have the sort of sum of a fixed constant energy and a variable energy. And the idea is we can then translate whether or not we're achieving a given threshold cutoff for activation in terms of what we think about in terms of T cell action goes. So activation can occur, for example, if I think, okay, T cell I'm giving by X and antigen by Q. If the interaction exceeds some lower threshold, the T cell's activated. And if we use this language or formalism, we can then describe other events that are interesting, like T cells survive thymic selection if the interaction between a self-peptide and that T cell is less than this activation for all of these self-antigens. And a T cell would recognize a foreign antigen if, for example, the interaction exceeds that for some foreign antigen on a threat, be it cancer or otherwise. Okay, and so we played around with so really the interesting part of this problem is in the constitutive relation that we assume to model this variable interaction, right? Because that's the part that's both a function of the T cell and peptide. And so we tried a lot of different things, and actually what we settled on was the following representation. So peptides are given by their sequence of amino acids. You have, a peptide sequence, you're given it, Kmer, where we think of K being on the order of 9 to 11. And the T cell now is going to be given by a random matrix of values where the ijth entry represents how a T cell, that particular T cell, would react with a peptide amino acid of species J at position I. So you have two things to consider. It's what variety of amino acid do you have, and what position are you in? And in this way, T cells can interact with positions in a position-specific way. So like if you have an alanine at position 2, that may be a highly desirable interaction for that TCR at position 2, but an alanine at position 7 may be binding with a weaker affinity. And so this is sort of the simplest, at least the simplest functional form that we could find that would sort of achieve what we wanted, which was position-dependent interactions. And I sort of have abstracted this as sort of different colors of TCR binding grooves that each have a different proclivity, if you will, for the antigenic amino acids. Okay, so maybe I show you how we would do this. So basically, the idea is that if you had a peptide, you can line this peptide up, and then the TCR is going to be given as this random energy matrix. And the idea is then that you would go at every position of this TCR that's binding this peptide, and you would essentially look for what that energy looked like, as a function of the amino acid. And then you would basically find all these values, and then you would sum them up. And these values in a toy model can be seen as IID, and they can follow some, generally reasonable distribution. We did this with uniform distributions representing min and max interactions, and we also did it for Gaussian distributions. The results are nearly identical via central limit theorem, actually. So this sum actually is what's interesting. Yes.
To clarify, so on the TCR receptor, you have these things there. So they can interact with any of those NWPs on the MHC?
Here?
Yeah. So anything can interact with anything across TCR and MHC?
Not in this model.
Not in this.
In this model, that's a good question. In this model, the amino acid that's at position 1 binds an abstract TCR binding groove in the neighborhood of position 1, and the local chemical environment at that location determines the proclivity for a good or bad binding event that's amino acid specific. In a model I'll show you in a refinement, you can consider this as an actual amino acid sequence, and then you've got non-adjacent interactions that can occur.
So that will get reflected in that matrix as lots of zeros somewhere, right?
Not necessarily, no. So, a good question. So this matrix is the TCR's desire to bind things at position 1, 2, 3, and so on. So the way we do this is we just take random, you draw random numbers for all of these, and when you actually evaluate or interrogate a TCR with a realization, you're only going to subsample a subset of them. Yeah, that's clear, okay. Yeah, good questions. Any other questions? Yeah. There's no, in this model, no. There's no assumed sequence dependence. There is evidence to suggest in some cases that there is some correlation in the neighborhood around which these antigens are more immunogenic if they have a doublet or triplet, but this assumes none of that. This is purely statistically independent selection. Yeah. That's correct. Yeah, so in this model, the only way you could account for that is by somehow having a uniformly lower binding energy at a given position across all amino acids to represent the fact that it's further away. We're choosing these energies IID though, so we don't account for that. You could account for it in that way in the model. We actually think there's a better way to account for what you're bringing up, which is the fact that some of these amino acids face the TCR binding groove and some are further away. So really, we should, in some sense, refine this model at some point to account for whether or not we have an amino acid directly facing the TCR, but not in this model. This is not yet. Yeah, good question. Any other? Yes. Yeah, that's another good question. So this assumes a single restriction. And so the only thing we do here is really estimate in our selection criteria that we're taking a subset of the peptides that are in thymic selection for a given restriction. And so it's, I think the estimate is something like 10 to the 5th or so self-peptides are, existing. And so we use about 10 to the 4th for a given MHC as an order of magnitude approximation. But presumably, yeah. So another thing here that's completely not described by this MHC has what are known as anchor residues. And so there is a very large restriction, usually at like position 2 and 9 or 10, wherein the antigen has to have a very specific kind of amino acid, maybe in leucine or an isoleucine, but it can't really deviate from that, for example. And so in those cases, that also ends up restricting a large proportion of the allowable binding events. Now, empirically, there are really good approaches, I think through Morten Nielsen's group, NetMHC Pan, and I think they're on 2.0, and they've actually done, I think, a very good job characterizing this problem computationally by saying, if you hand us a restriction and a collection of antigens that you're interested in, we can confidently give you a distribution and underlying what the likelihood of binding or interface might be to get it to the surface. But what we currently lack are models that describe if you hand somebody an MHC restricted T cell, with an antigen that binds that MHC, does it react or not? We know something about the T cell, we know something about the peptide on the MHC, but that's sort of a more nebulous question. Which motivates this? Yeah, good, thank you. Any other questions? Good. Okay, so again, back to kind of the toy math modeling. So we can do things, we can simulate thymic selection, basically. We can take on the order of 10 to the 4th self-peptides presented on this MHC, and then we can take T cells, and we can ask, okay, if they're binding all 10 to the 4th of these, the T cells that survive have to be the ones that don't bind above some activation threshold. So you can plot the percentage of the T cell. This was called the Rice model because we developed it, I guess, at Rice. And this is Rice blue, which contrasts with the A&M magenta now. But as a function of activation threshold, the percentage of T cells that are deleted centrally declines, right? So here, the T cells don't have any ability to react because they require uniformly strong binding to even activate. So there's no deletion. And here, even a minimal amount of activation results in a T cell activating. And so if you take T cells and you subject them to 10 to the 4th IID interactions, essentially all the T cells get deleted. And so there's this sort of middle ground. This is actually the probability that this interaction is greater than E sub n for all of the E sub n. This is an extreme value, a Gumbel distribution. And we can do the math, and I'm saving sort of the mathematical details for the sake of time. But what this allows us to do is it allows us to say, okay, in our abstract math model, how do we choose this threshold to physiologically replicate thymic selection? And we can choose it, actually thymic negative selection's survival percentages are estimated anywhere from 20 to 70%. It's actually very less understood, wild variation in the belief, but we choose a 50% cutoff. And from that, just like we draw a selection curve, we can also draw a recognition curve. And so these slightly busier curves are now the same x-axis, the T cell activation thresholds. And there are two plots. The left plot is the probability that a T cell conditional on surviving thymic selection will recognize a foreign, unrelated peptide that has nothing to do with the self-peptides that the T cells were trained to avoid, right? There were 10 to the 4th of those, but now I just choose a random peptide, and as long as it wasn't in the original list, then this is a foreign peptide. And then on the right, this is the probability that a T cell can recognize, conditional on surviving thymic selection, a tumor-associated peptide. So the rule there is you choose one of the self-peptides that you know the TCR couldn't recognize because it survived thymic selection, and you mutate it in a single position. And the question is, does that get recognized? Now, when we did this, we thought, of course, these recognition rates have to be very low relative to foreign because the T cells were trained not to avoid recognition of self, and we're just mutating it in one spot. Now, okay, there's a solid line for each of these, which is our analytic theory, and then we have simulations. And so I want to draw your attention to sort of the red crosshair, where the simulations converge to the theory in both cases. This is where thymic selection that gives physiologic selection rates is operant. So in this regime, if we compare these two graphs, we see the foreign is recognized at much higher probabilities than the point mutated antigens. But really, the difference here is very small. The sort of range of this plot is the same. So this, to our surprise, suggested that simply mutating one amino acid position away from self, which the T cell recognizes with probability 0, right? By definition, it can't recognize self. It was trained to avoid it. One mutant away gives you about 75% of the recognition probability that you get by choosing a completely random antigen sequence. It has nothing to do with self. So this was sort of counterintuitive and surprising to us, and actually, in fact, supports the robust recognition of tumors via point-mutated neoantigens. And in fact, subsequent work has sort of showed that this depends on more complicated things, but in fact, neoantigens can lead to durable remission outcomes in many cancers. Most notably, perhaps, would be melanoma. Yeah. So, yeah. So if I go back, this sum is a sum of IID in theory. IID uniform 0-1 random variables, so we have 10 of them. So the support, the range of the random variable we're modeling is 0-10.
Yes.
Okay, so that's here. And what we do is we just basically can analytically or numerically, same result, we can plot the percentage of T cells that would die if the activation threshold were chosen at E sub n ranging from 0 to 10. And when we choose it at physiologic selection, we have a criteria. Yeah, that's a key part for this, because otherwise we have no connection to reality. Yeah. Does that answer yeah. Thank you. Okay, so moving on. Yeah. NN. Sorry, this is the number of self-peptides. And so the blue one here is physiologic. There's about 10 to the 4th self-peptides for a given MHC restriction estimated in the thymus. And so really where your eye should be is the blue, oops, whoa. That got it away from me. The sequence of blue plots can, in this neighborhood of about 8.15, I think, is about where we're looking at each of these. And so our theory is a theory that's idealized for a smaller number of peptides in the training step. Yeah. And these are all based on probabilistic arguments. Okay. So as a few people have pointed out now, there are simplifications in that model. One of the simplifications was that the TCRs were idealized binding grooves. Well, we can idealize all we want, but if we want to compare to real data, we need sort of a tangible way of representing a T cell as something that we can, again, compare directly to data. And so, and that was one thing. So the first step is to represent TCRs as primary amino acid sequences as well. And the second step is there are non-adjacent interactions that occur by amino acids on the CDR3 of the T cell and the peptides that are loaded in the binding groove. And so, in this next sort of iteration, we took care of both of those problems, and we really used crystal structures of published T cell peptide pairs, which gives you the three-dimensional sort of alignment or configuration of this binding event, from which you can then take the distances between all pairs of amino acids in this interaction, and you can plot it or represent it, and this is the way I like seeing it. I call this a contact, or we call this a contact map. So this is really a sort of a readout on at what positions in the antigen in T cell contacts are being made. Another thing I want to mention here is until this time, or around this time, all of our theoretical models assumed a diagonal contact map. including ours. I mean, the one I just presented to you assumes that you have a one-to-one interaction between amino acids in the peptide with the TCR. And so this really, I mean, this is data derived. This is actually a real example from a real, this is a 3QIB, so this is mouse, this is a mouse, actually MHC class 2 model, so very different than human. But nonetheless, humans also have similar behavior where you have this highly non-diagonalized model, and that really affects the way that we would estimate those antigenic sort of recognition rates. Okay, another thing, so I want to say that when you do this, you can then very quickly appreciate that there are going to be some subtle differences between contact maps involving different T cells and involving different antigens. And in fact, if you change any one of those, the contact map is expected to change. So here actually, this is a family of contact maps that look largely the same. So they're all the same MHC restriction. So that's the first kind of prerequisite for assigning similarity. And the other thing is, these two share a common antigen. The TCR, the T cell is different. And these two share the T cell with an antigen being different. And so we can vary one or both of them, and we can see that they sort of manifest in subtle ways in the contact map. The other part of this is that the contact maps vary a little bit, which gives us ever so slightly different proclivities for these particular interactions to favor or discourage selection, and also recognition, which I don't show here. But this family of curves here, you can see they kind of, this is the idealized diagonal model, a selection curve that I just showed you, and this is models that are non-adjacent, so they have higher variance, and they have a little bit of variation depending on the contact map. And so this tells us structure matters for the problem, and even the likelihood that individual TCRs would be selected for and maybe go on to recognize antigens depends on the structural topology. I think I have, yeah, so I also wanted to say that really here, what matters is the energy, which it's stated a little bit differently here. This is E sub C as before, plus the variable interaction that we talked about in the last couple of slides. But now what we need to do is we have to have an energy between two amino acids, and we have to weight it by how much did they have contact. So it's two factors. Are those amino acids repulsive or are they attractive? Okay, that's one part, and that depends on the amino acid at a given position for both sides. That's a pairwise interaction. And then how much contact did they have? 0 to 1. Multiply that and add it to the matrix, or the overall, I should say, overall contribution to energy. The energy matrix is used to derive these values. So very recently, actually, with a talented postdoc, we have been working on getting an exact answer for the variance of any arbitrary contact map if you specify this and you have any distribution of amino acid energy matrices to be assumed. Before this, we had to rely on approximations, but we're using probability arguments to characterize variance, which then goes back to the question that matters, which is, are these likely to recognize something? Because the variance is what you care about if you're asking on a collection of, a large, number of antigens, what's the extreme value that you're going to pull? And so that's more related to variance. And we can actually do this. This is just to show you. We've now analyzed a lot of systems. These are all human systems that I'm reporting here. And I've plotted our analytical variance calculations for idealized motifs. So this is the diagonal motif that all of the theory had sort of used before. None of them follow that. And this is an idealized box motif where you just scale the number of contacts in the box. And you see that actually a lot of them look maybe sub-sparse, but they're close to a box. And then some of them are enriched above the box motif. And so we're really interested in understanding fundamentally at a systems level how the sort of deviations in this idealization from a box, ideal box, or what have you, these sort of excesses and deficiencies of variance will, sort of at a holistic level, allow one of these to recognize antigens easier or more difficultly in nature, right? We only have a subset of these sampled. The crystal structures are sparse. We don't have many of those, but we have many examples of antigens. So at a systems level, how do we predict whether or not one of these systems is going to have an easier or harder time? This is variance over realizations of say, for example, peptides that could be presented in this MHC. So I would motivate that thinking with, if you have a viral threat and you have a repertoire of viral-associated antigens that are being presented, they're non-self, then the question is, can your T cell recognize at least one of them? And so the question is, if you plot over some distribution of viral peptides, the binding distribution, the ones that have higher variants are going to have a higher likelihood of at least one binding. And so this variance is sort of a surrogate measure of how sensitive can these things be to the recognition of a repertoire and a small ensemble of threats as a function of the number of contacts at the interface. So, presumably some of these have more contacts, some of them have less. We would have to kind of consider it as a normalized to number of contacts. Yeah. Good question. So I think this is all the random energy modeling. I now want to move kind of into, practically speaking, if you have this model, what can you do with it and where do you want to go with it? What we really ideally want to do is we want to take real examples of T cells and peptides that are provided to us, maybe examples and non-examples. So we have good binders and weak binders. And we basically want to say, given a structure and a set of these strong and weak binders, can we train a pairwise energy model so that we maximize the energetic difference between strong and weak binders? If we can do that, then we can try to evaluate, given an unknown list of TCRs and an unknown list of peptides, what their specificity map is, which is that's the big unknown currently. And we've only partially succeeded, as well, as you'll see in a minute. But really, the idea would be, patient comes into the clinic, maybe you predict their tumor-associated antigens, you have a list of 1,000 of them, maybe 1,000, and then you have T cells that you could target with these antigens. You have to pick out which T cell would be most likely to recognize which antigen. So the antigen list is person-dependent. The T cell list, if you're doing adoptive T cell transfer, can be chosen out of a candidate list of T cells that are realized. But you really have to say, I have these antigens. Can any of them be recognized at all by T cells that I want to introduce to cure the cancer? So that's sort of this outline. Now, this was work that was done in mouse class 2 MHC because we had more structures available at the time. We actually have most recently done this in human MHC class 1 for HLA restrictions to the most common HLA allele in humans, which is A0201. So this is to say the MHC molecule is fixed, and we're only considering right now describing those interactions that occur on the most abundantly expressed allele, for which we have the most abundant structural and actually sequence data. So this is kind of busy, but the idea here is that we train, again, strong and weak binders, T cells, peptides, that we know either they're a good match or they're a poor match. And we have now a family of structures, because there's about, I think, 66 total published structures for this particular system. And we have to feed in, using the best structure, those TCR and peptide pairs to do the same optimization of strong and weak binders as I mentioned before, optimize this pairwise energy matrix, and then, given an unknown T cell peptide sequence, come up with a good way of finding which structure best represents that sequence, because we're limited by structure, threading that sequence through the structure, and then making a prediction. And the hope would be, if we do this, we could guess whether or not we have a strong or weak binding TCR peptide pair. Does everybody follow that general outline? Okay, so this is now just sort of an illustration of how structurally diverse these things can be. That we didn't have this problem in mouse, actually. When we looked at mouse, and we looked at the MHC IE of K, the class 2 restriction, the structural similarity was very, very similar, and we were able to predict very easily. When we did this on humans, we tripped and fell, because we weren't accounting for this diversity. So what I'm showing here is a hierarchically clustered plot of structural similarity for all of the available crystal structures for human MHC class 1 HLA A02. 66 or so of them. And all they've done has been clustered by structural similarity. Then, separately, you can overlay the biologic or functional significance and appreciate that groups that cluster similarly are coming from similar functional groups. Sort of We would hope that's the case. I mean, if the heterogeneity is such that, MART1 antigen looks like TAX or NLV, we have no hope of doing this. So, okay, so that's point number one. But point number two, you can even see that in some of these systems, a remarkable amount of kind of inter-system heterogeneity exists. So it's not even sufficient sometimes to use one structural template to describe everything in a similar functional group. Okay, so now let's say, how do we do if we do this on sort of cross-validation? So what I'm showing you here is now the predictions of this model. If we train it on about half of our data, and then we cross-validate using a holdout of that data, where we know that the answer is a strong binder. So these are examples that we know interact favorably via binding. They have strong binding affinity. And the gray here is the distribution of null binders. So these are all where weak binders are. This is binding energy. So a higher binding energy is a more destabilized or weaker binding, and a lower energy is better. And the predictions are given by these points, either open or closed. And training is given by open, closed is testing, and if we did well, it's green. And if we did poorly or really poorly, it's brown or black, respectively. So you can see that for the most part, I mean, we want these points to be very resolved from the distribution of null binders, and we see that for the most part we do that. There are places where actually this model fails outright. So in this particular case, I think this is NY-ESO. It's a one particular antigen. We can't do it. We don't do it very well. And then in sometimes, yeah, we do mostly well, but then we miss on occasion. So now this is data that looks similar to the training data because we took a subset of the training data, we held it from the model, and then we tested it. But the model still is looking at similar data. So we next asked, can we predict on data that looks nothing like the training data? The only thing we want to make sure is that we're using HLA class 1 A02 restricted, just like before. And so there are actually several good data sets that came out. One most recently was from 10x Genomics. And so they published this affinity description of TCR peptide specificity, which involved about 1,700 TCRs and then 22 different peptides. And so the only results I'm showing here are results where we removed the TCRs that were shared in common. Any TCR that was shared in common between our training or our cross-validation set was explicitly omitted from this analysis. And then here we're actually, I now switched the graph on you. This is Z-score. So a really good prediction would be that if all of the TCRs that are in this 1E6 group are predicted with high Z-scores, because they're all strong binders. And a poor prediction is if it's below this cutoff of Z equals 1 that we use. And you can see that for the most part, we do reasonably well on many of these cases. And there are some places that we actually fail outright, right? These are distributions that we really are having a hard time accounting for. If we go back to the structural similarity, that is, if we ask how similar are these to sequences that we have a known structure for, there are places that we have no good sequence. And in fact, those are places where I've put this purple asterisk. So actually, a lot of these correspond with cases that we just don't do well on, which really, I think, indicates two things. In our model, you really need a good structural template to do well. And the structural template that we're using is actually generating the predictions when we were able to accurately characterize the system, because we don't have a lot of training data. We have on the order of 100 pairs that we're training, and we're trying to infer in a space that's very large based on my maybe first slide's order of magnitude estimates. So, okay, so this is sort of, this is what we've done. Now, more recently, another task that I think is a really good task is the following, which is in the clinic, so at MD Anderson, for example, for this one, they will have antigens that they know are expressed in the body of a cancer patient. And in AML, for example, when the patient is immunosuppressed, T-cell expansions can either represent a desirable expansion against a tumor antigen, or they can sort of represent a distraction, which is an expansion against a viral reactivation event. So when in the clinic they take the T-cells that have expanded, they really want to know which ones recognize tumor and which ones don't recognize virus. You don't want to give the viral reactivation ones to a cancer patient. So here, what we did was we took our original training data set, which was what I just showed you, and we took a little bit extra TCRs that our clinical collaborators confirmed were specific to flu, which is an influenza antigen, as well as NLV. This, I believe, is a cytomegalovirus, again, another reactivation-specific antigen, as well as MART1, which is a tumor neoantigen. And the goal was, okay, just as if they perform this test, they know those T cells recognize something. What did they recognize? Was it MART1, was it flu, or was it NLV? And our test data was 120 of these with that we were blinded to with confirmed specificity against one of these three. Okay, so we actually, this sort of seemed like a pretty straightforward task to us, which is, why don't we just in silico do what they do, which is they have an affinity-driven process where they pull a TCR out. The TCR bound one of those three antigens, but it didn't bind all three of them strongly. So they just don't know which one pulled it out. So we do the same thing, and we say, well, we can test that T cell of unknown specificity against MART1, against flu, and against NLV. And hopefully, that T cell is a strong binder for one and a weak binder for the other two. So what we do is we train our model to understand the fingerprint of what a MART1 strong binder, but a MART1 weak binder to flu and a MART1 weak binder to NLV would look like. And we do the same thing for flu and NLV, vice versa, so that when we get that triplet of predicted z-scores, we can then match it to a fingerprint that's either more consistent with a MART1 strong binder, a flu binder, or an NLV strong binder. And so when we do this, actually, I just point this out, that we were having difficulty because MART1 is actually very diverse in the sort of bimodal distribution of strong binders it has. And in fact, we found that this overlaps exactly with the structural differences within the MART1 subgroup. And so MART1 weaker binders, although they're still strong enough to elicit a response, are coming from this structural cluster, and those that have a more strong interaction are coming from the second structural cluster. And so we actually had to account for that in this calculation. And so here's the, here's the sensitivity, specificity, diagnostic accuracy, and the ROC curves for this test when we did it on unseen TCRs. And you can see that we're doing actually reasonably well when we have about 50 new data TCRs to incorporate into the model. And so we're sort of pleased by this. And I think the next step is, what's the limit? I mean, can we do this for 10 peptides? Can we do this for 15? Every time you add a new peptide, you have another family of weak binders that you have to resolve. And so these distributions probably have some fundamental limit, but we're currently still exploring this. Okay, and I think for the sake of time, I'm just going to move on, but I just want to point out that another task here is point mutants. It's very hard to understand if you point mutate an antigen, like a tumor antigen, is it going to make or break specificity of a T cell peptide pair you already know? Will it enhance immunogenicity? Most of the time it won't. Most of the time, if you find a strong binder and you alter something, it'll break the binding. Sometimes it enhances the binding. So we can sort of replicate this on known experimental data where the purple, this is a whole comprehensive point mutagenic screen of this particular peptide, where they point mutated it in all possible positions and all possible amino acids, and then they put in pink here where immunogenicity was broken. And so you can see this is our kind of paler pink version of this that we reproduce pretty well using that type of model. Okay, so are there any questions on TCR peptide specificity prediction before we move on? Yes. Yes. Absolutely. So in this model, we're using the structural framework where we have a contact map. So what we find is these, I mean, it should be of no surprise perhaps that this sort of central region is the region is the most contacting region of the T cell. And therefore, those are the amino acid positions that if the virus or tumor were to evolve and create a variant, they're gonna be successful if they break that immunogenicity. Yeah. The energy matrix for the...
So that's a pairwise amino acid.
Yes, yes.
So is it necessary that has to be based on only PCR and instructions, or possibility to
Yes, yes. That's a very good question, and I don't think we have the answer to it. What we do know is that if we, so this just describes what's the binding affinity or interaction between a glycine and a, I don't know, a glycine or a tyrosine. I don't have all of them memorized. So some of these interactions are strong, some of them are weak. There are actually canonical, famous, the MJ Miyazawa-Jernigan pairwise amino acid energy matrix, where physical chemists have actually directly tried to estimate this interaction in isolation in chemistry experiments. What we find is the energy matrices that we optimally find that are capable of resolving TCR and peptide interfaces are not the same as those, meaning that we really have sort of a residue-specific interaction that really helps us to make predictions. I don't think that we have different physics going on. It's not like the amino acid interactions are suddenly different in our context than in any other context, but I think because this is a coarse-grained model, this map really represents other things like size, and with the proximity of amino acids, we're not accounting for kind of higher order effects. We're just talking about energy. So really we're training it as a group of 21 choose 2 parameters that we can try to align with. But I think the victory here is we can do that with one energy matrix for a very large system of cases. Yeah, good question. Okay, so then let's see. Maybe I'll continue. I'll do the 90 minutes and then we'll pause for, we're about at an hour now. So I'll go into the second part of this, which is tumor-immune coevolution. So, okay, so a little bit more background. So this is a kind of now famous picture of, I think this was AML. This is a liquid tumor where, sort of you have these stem cells, you have sort of described here as an expansion of the tumor, it grows out, and then there are mutations that can occur that when you start giving chemotherapy, some of these mutations can actually confer resistance to the chemotherapy, and then of course, unfortunately, despite the continued administration of chemotherapy, you have subsequent drivers that sort of explode. So this is the problem I think that we've all seen with cancer so far. Now, this makes cancer treatment very difficult. From the perspective of the T-cell repertoire, as I mentioned in the beginning, though, we have a targeting mechanism that isn't one fixed target. It's not like you're giving chemotherapy and then the cancer gets resistant to chemo. You have a T-cell recognizing an antigen, and that cancer might lose that antigen, but it might create a new antigen that can be recognized by a completely different T-cell, and so on and so forth. So from a dynamical perspective, you can have repeated recognition evasion opportunity. However, we know that that doesn't always work, and there are similar evasion mechanisms that occur in cancers as well. And I want to motivate the next part of this talk with a very kind of, it's rare, but it's a catastrophic evasion mechanism, which is loss of a diploid MHC. So oftentimes cancers can downregulate or mutate away presenting antigens on one of those MHC molecules. And if that happens, and that's the only way the T cells are able to recognize tumors, you can forget that there's going to be any hope for them to recognize the absence of a new antigen being presented. Okay, so this is kind of a toy model now. So, and then I'll sort of refine it a little bit. But the way that we think about this very, very simply is that there's some cancer dynamics that are growing. This is a continuous time birth-death process. Some net growth rate are, if the immune system can recognize the tumor, it sort of incurs a death rate D. The immune system can expand, and a lot of T cells then drive the destruction of a tumor. And so this death rate is, in theory, greater than R. So if it happens, you can drive disease elimination. And that would sort of be the hope. But of course, cancers can mutate too. And so we have this mutation rate that we are thinking of as a rare but catastrophic loss of recognition ability. And this is a per cell mu division rate. And the other thing too is that for reasons that are not completely understood, there is a minimal theoretical limit upon which the immune system can recognize any threat that's dynamically growing. And so we have some lower size limit of detection. So the way this process evolves is the tumor starts growing. It reaches some critical lower detection size. The immune system can now interrogate it. And the immune system, we sort of model with two pieces here, which actually relate to what we spoke about a moment ago. But one of these is the sort of attempt to describe the total T cell repertoire diversity, which is the number of available T cells that the immune system may have available to it when this becomes a threat. And the other part of this is an immune turnover parameter of new immune cells coming into the region. Maybe if this is a very inaccessible region, this could be inherently the characteristic timescale for T cells to arrive. If you have thymic selection operating, this is also like an output of T cells in thymic selection. And so, okay, when the tumor gets this size, the existing repertoire can recognize it with probability P sub C related to the diversity of T cells you have. With probability 1 minus P sub C, it doesn't recognize it. And if it doesn't recognize it, there's a continual opportunity of new T cells to arrive according to this Poisson arrival. Maybe there's a recognizing T cell that arrives that can clear it. If no recognizing T cell arrives, the tumor escapes, and that's a stopping condition. If the tumor is recognized and the T cell responds durably, then there's elimination. This is what we want. And then we have this possibility of an acquired immune evasion event where somewhere along this growth and killing trajectory, an immune evasive phenotype, which I've kind of given as this ghostly red, in this picture cell emerges, and there's no hope for T cells to recognize it in this model. So that's sort of the basics of the foundational model. Now, what we can do is we can ask things like, for example, if I just describe this model as a stochastic process, there's a probability of escape and elimination on the tumor's part. And in particular, what's the probability that baseline your immune system is able to clear the tumor? That's given in this very kind of pale dotted black line in all of these curves. In these that probability of killing the tumor is given as a function of the relative growth rate of a tumor relative to that killing rate of the immune system. So most tumors can't grow as fast as the immune system could kill them if the immune system robustly responds. So they should all be this should all be 0 to 1. And then I've also, going from left to right here, I'm plotting sort of physiological declines in T cell performance. So first we have, compromised thymic turnover of T cells with a preserved diversity. And then eventually in sort of more advanced, age, actually, you have declines in both turnover and diversity. And so we can ask questions like if we, have a CAR T cell that's specific to a particular antigen that's being presented by the tumor, can that CAR T cell recognize and recover elimination? How much better does CAR T cell versus neoantigen vaccine therapy do above the baseline ability of the immune system? So this is what the model can do. Now, one thing that and of course, if we wanted to sort of put this in a very particular disease, we would have to have more constitutive models to exactly understand the relative abundance of those antigens that CAR Ts can go after, and so on and so forth with the vaccines. But what we were actually really interested in is that this idea that it seems like the recognition capacity of the immune system naturally is very dependent on the status of how healthy the immune system is, if you will. Like, is it very diverse? Is it not diverse? Or is there still turnover? Is there not? So what we really wanted to do was we wanted to say, how do we think about the risk of getting cancer just as a consequence of an aging immune system? And so one way to do that is to choose, in this case, AML, because, we like AML, but AML is a liquid tumor. Tumor accessibility isn't as much of an issue. Yeah. Yes. Say that again?
The y-axis is the probability.
That's correct, yes.
So is it odd that the probability goes down?
No. So it may be curious. Actually, there is this phenomenon known as sneak through. So slowly growing threats can sneak through an immune recognition. So actually in tumors, we sort of see this. Like very rapidly growing tumors can get how do you say it? Capably cleared. Also, very indolent tumors or intermediate tumors can get very well cleared, and then you have this sort of non-linear behavior that as you vary the immune system, you get, or vary the growth rate, I should say, you get behavior where fast and slow growing tumors behave similar. So clearance is, this is clearance probability. It's maximized for intermediate threats. So you have, I guess what you would see is, maybe I said it backwards, but it's the control of intermediate growth threats, and then kind of the skipping out of high growth and low growth threats. why does this happen? I mean, in our model, we actually have a phenomenological reason. The reason is because if you go back here, just to use the illustrations, this minimal size is growth rate dependent. There's this growth threshold conjecture that says that the rate at which the antigens are being produced is what matters to the immune system, not the size. So if your growth rate is smaller, all else equal, you're going to have to wait a longer time for the immune system to even recognize the threat. That longer period of time actually allows for immune evasive clones to come. And then, if the threat's recognized, it dies a speedy death. On the other hand, for very fast-growing threats, which I'm not showing here, they get recognized quickly because their threshold is low, but then the battle between the immune system and those threats is very fought out because the difference between growth and death rate is not so high. And so they survive long enough to hopefully give rise to a clone. Yeah, it's a really good question. So this is called, if you want to know more, this is called sneak through. Yeah, it's an interesting phenomenon. Okay, so the question is the following. If you have a cancer incidence curve that's age specific, can you take physiologic declines in T cell turnover and repertoire diversity and do a reasonable job predicting this behavior? So this data, again, is leukemia because T cell accessibility is something that we don't model. I mean, this is a well-mixed model. We don't want to think about three-dimensional T cell topology. And we have good data on AML incidence as a function of sort of more advanced ages where T cell declines occur. So what I'm showing here is our theory where all I've done, I mean, there's one variable that we can vary. The variable is the baseline rate of tumor initiation for all ages, constant for all ages. We assume that it's no more or less likely that you get an AML clone at 8 versus 80 years of age. And that is a free parameter that we use to fit this model where we build in age-related declines in T cell repertoire and diversity and turnover. And we actually do a reasonable job with our one parameter fit. So then the question is, how does this do compared to other models? I mean, perhaps the most famous model is an old model. It's the multi-hit model of cancer incidence, and it assumes that there's a statistical number of independent events that have to occur before tumor initiation and propagation occurs. So what I'm drawing here is a family of the best fit of all of these n equals 3, 4, and 5, 3 through 6 hits on AML. I think AML has, it's believed, maybe 3 to 4 hits. And you can see that, I mean, these are best fit lines as well. So our model can kind of characterize this like decline in the incidence that caps off at the end, which is actually due to the saturating effect of repertoire diversity shrinking and reaching a sort of bottom point. Whereas these models, I mean, the multi-hit incidence models are all monotonic. So they sort of assume that as you live to be 100, you have an exponentially larger, arrival of tumor. And I mean, it's interesting because I think here there's some reason to believe that maybe if you live that long, you may have an immune system that's intact. So we by no means want to throw out the multi-hit model. I mean, this is a very relevant model, but I think the important thing to realize here is this really advocates for including immune parameters into sort of our epidemiological modeling of cancer, right? Because this is just based on physiological declines, nothing more or less. Another interesting thing is that the cancer incidence rates are, for cancers that are not sex dependent, usually higher for males than females. You can build in the thymic repertoire diversity and turnover rates, and females have higher repertoire diversity. And so it's actually an interesting exercise then to repeat this and see the sex-specific difference. But I'm avoiding that. Yeah. Yeah. So this is noisy. I don't remember this. So this is from cancerresearchuk.org. This is just this is a public repository of incidence rates published through the UK. I don't remember how many were in this group, but you have to think of these as being pretty noisy in the. I would not say constantly increasing. Yes. Yeah. I mean, I think, if you look at this, I mean, okay, I mean, probably the data sampling in this region is limited, but I mean, it's a countrywide study, so we probably need to look into how many there are. But you can tell that it's certainly it's non-decreasing, but I would say that the rate of decreasing is sort of, I mean, this looks more like a logistic growth behavior to me than exponential. But yeah, I mean, certainly the noise here is higher, yeah. That's right. Yes. Yes, yes. Yeah. Right. They're normalizing this to 1 in 10 to the 1 out of every 10 to the fourth, or negative fourth is how I plotted it here. Right. Okay, good. Yes. So yeah, good question. I think the answer is it's complicated because, so for example, AML is not a highly mutagenic tumor. It's actually very, I mean, the mutational burden is very low. So, right. And this is why we chose it. Now, the quality of antigens, the abundance of antigens, I mean, and the tumor-immune microenvironment, like these things are hugely, we know they're hugely important, and we don't model any of that here. In part, we think we can get away with it with AML because of the well-mixing assumption. But in general, yeah, those things all matter and would presumably refine such a prediction. Yeah, great comment. Okay, so we did this. So the next thing was, we have this idea that we have a model that we've now, we had one free parameter, and we think we're explaining AML incidence well using this model, using only age-related declines in immune system's ability to fight cancer. So the next question was, are there cases in the real world where patients suffer an artificial immune decline over large periods of time? And if so, this model would absolutely suggest that their cancer incidence rates are higher. And so, number one, does that exist? And yes, we would predict that. And number two, how well do our predictions agree with data on that topic? So we thought a little bit about this. And in fact, what we ended up converging on was chronic solid organ transplant recipient patients. So these are very sick patients. They come in, they get a transplant, but they're put on T-cell immunosuppressive regimens for decades just to prevent their organ rejection. In a similar way, graft versus host disease, as we talked about before. So those patients are being prevented by giving T-cell inhibition therapy. And so the question is, if their T-cells are being inhibited for decades, can we predict the relative enrichment of AML? So that's the last point that I want to make here, is that on the left is our prediction, and on the right is sort of the reality, where the only thing we do here is assume mild declines in the T-cell repertoire pool, but we keep all of our other parameters that we fitted the same on the other slide. And so what you can see is, yes, in fact, at least as an order of magnitude, I've normalized the y-axis here the same, and then I've kind of given you the numbers here, that we are in the correct ballpark of being able to predict this at a 10-year horizon following chronic T-cell immunosuppression. And so together, we sort of are concluding not only age-related declines, but also iatrogenic or clinically induced immunosuppression together explain increases in AML incidence, which again, sort of impresses upon us the reasonableness of including an adaptive immune system in any model of cancer incidence. Okay, so I'm going to try to keep on time. So let's see where we can get in 10 minutes. One of the simplifications here was we assumed there was one MHC mutation that just completely made the thing invisible from the immune system. If you had that mutation, there's no way the T cells can recognize, no matter how good they are or how diverse your repertoire is. That's a strong assumption because that's a very rare and catastrophic durable evasion event. In reality, though, things can happen like tumor antigens might be downregulated. So you don't see that antigen anymore. Or maybe the antigen's mutated further and then the T cell can't recognize it. So these are more transient, less durable evasion events. So really what we wanted to do is we wanted to take this model and we wanted to say, if this evasion event isn't so durable, we can have the same process repeat over and over again. So this was, again, a continuous time model, stochastic model. As it turns out, for technical reasons, it's very hard to sort of solve for things like escape and elimination if we kept this in continuous time. So we instead retreated into a discrete description of this where we think about each of these events as being a generation where you have branching that's occurring. So now this is sort of a discrete time stochastic process where you can describe things like a branching parameter that is only dependent on the killing rate, the birth rate, and then these mutation minimal detection size that you have. So we have this, sort of given by the previous model parameters. And then the probability of clearing a new thread is the same. And in fact, you can sort of solve for the fixed point of this transformation to come up with the long-term elimination probability. And so I'm giving simulations here in red and blue, or red and green rather, of the long-term elimination probability and then the analytic theory, and you can see agreement there. So we can do this where we have repeated recognition and evasion. I actually think this is a much more fun depiction of it. So this is really one single kind of cancer idealized in our model. The initial conditions and the parameters are all the same, but the stochasticity is the only thing that's different here. And what you see is sort of this cat and mouse chase between an adaptive immune compartment capable of repeated recognition events, and then you have the tumor that has repeated sort of escape or evolution events that lose that recognition. And that battle can occur, perhaps for a short amount of time prior to escape or elimination, or maybe a very long time. So it's a very, stochastic-driven model, I would say, which is characterized by very large waiting times. And in this kind of intermediate state in the tumor-immune world, we sort of think of this as kind of like equilibrium. So there's this concept of 3 E's of immunoediting. You might have, elimination occur. There may be some equilibrium if elimination doesn't occur, and then ultimately after equilibrium, you get an escape event. So this is just the readout of our model in that sort of way with individual trajectories. Okay, one thing we could do with this model, for example, is go back to the clinic or our clinical collaborators, and we can ask questions that we think might be relevant from understanding repeated immune recognition and evasion events. So this again these are I believe this is CLL now, but these are responders and non-responders, and their distributions are given for this measure of cellularity. So cellularity in their world can mean the number of unique clones you have in the tumor. If you have very distinct clones, you have a more heterogeneous tumor and your cellularity is low. If you have one dominant clone, your cellularity is high. And so our question was, can we take our kind of repeated immune coevolution model, and can we basically try to identify places where we would recover the probability of a non-responder and a responder? So we have a few patients that don't respond and a few that do respond. So we have a probability of response. And the question is, if we figure out where in our parameter regime that probability of response is, we get some sense for the parameters that might characterize this population. Then we can take that parameter that we fit as fixed, and then we can simulate our in silico patients and say, okay, number one, can we derive the adequate probability of response? That's a sanity check. We hope we do, because we're choosing the parameters in that regime. And number two, if we have non-responders and responders and we sort them out using our model, can we recover the distribution of number of clones for each of the responders and non-responders? And so when we do that, I'm sort of, let's see, this is non-responder here, non-responder, and then, so red goes with red and gray goes with black. And so we do see, in fact, that, and perhaps it's not surprising, there should be an enrichment in responders, the ones that are getting T-cell therapy, to be ones that had a more clonally homogeneous cancer. The ones that have very heterogeneous disease are going to be ones that are harder to treat via this sort of T-cell population dynamical model. And so this is about the furthest we went with population dynamical models to explain this. This is, again, T-cell transplant. They have other kind of separate treatment modalities that would then be further incorporated here in distinct models. But we're encouraged at least that we can recover the distributional responses if we're looking at clonality, for example. Okay, and then maybe the last thing I do before we break is just mention that all of this happens in solid tumors in an immune microenvironment that has structural topology. And so where we're going with this is actually trying to repeat describing the immune tumor coevolution in a solid tumor and not a liquid tumor. And in a solid tumor, we have spatial examples of how tumors evolve and what their spatial distributions are, in addition to the spatial distribution of the T cells that are interacting and presumably recognizing those tumors. So the thing that ends up actually being kind of reported a lot in the literature is this tumor-associated collagen signature, or TACS. And so sort of in looking at tumors, there's this sense that there's a TACS1 that represents a random orientation of these collagen fibers. TACS2 is sort of associated in many cancers with intermediate disease, which is circumferentially aligned collagen fibers, and then TACS3 is radially aligned into the tumor and usually associated, at least in breast cancer, with more advanced or metastatic disease. And so our questions are along the lines of, can you look at these TACS configurations, build them into an agent-based model, and get a sense for the way in which the spatial distribution in which tumors evolve, as well as the sort of spatial way in which the immune system can interact with those tumors that are evolving. And yeah, actually, it's not quite clear. In some way, if we look at the way that TACS2 and 3 help tumors versus T cells, TACS3 is the one that's usually associated with higher aggression and more advanced disease. But we actually find that T cells, in theory, in that configuration should much more effectively hone in on tumor. And so for the sake of time, I didn't want to get into the data here, but our sort of early model is suggesting that you need more than just TACS3 in order to recapitulate survival curves that are seen in the clinic that suggest TACS3 is more severe. And in particular, you need phenotypic events that change, like EMT, for example, and less immunogenic tumors to explain more aggressive TACS3. Okay, so I think this is a good stopping point before I'll finish off the research talk maybe in the first 10 minutes of the lecture. And this is a good segue into dynamic programming, which I hope everybody's interested in learning about. But we'll start here and then end with the teaching. Thank you.